Good afternoon. Just coming up to the start time, so we're just going to wait a few moments and give people a chance to log in. Whilst we're waiting for people to join, just as we're starting off this webinar, if you're logging in, could you please let me know if you can hear? So just in the chat box, just say yes or hello or something. It's just really handy to know that the audio is working all right and that you can hear us. Great, somebody said yes. Fantastic, thank you very much. Okay, so we've hit 1 o'clock. It normally takes a couple of moments for everybody to log in, so whilst we're waiting for that to happen, I'd just like to ask if you're happy to, can you pop in the chat and let us know where in the world you're joining from? It's really interesting, just as a fun thing for us and out of interest to know where our audience is. First person is from London. Well, I hope you're enjoying a lovely sunny day. I managed to get outside a little bit, so that's really good. So we've got some from China, Sussex, South Korea, quite a few people from London, somebody from Istanbul. So I can see somebody from Malaysia and Indonesia, Singapore. So it's just really interesting for us as well. Quite a lot of people from London, which is really interesting, and Leeds. So thank you very much. That's really helpful. It helps us to also to know what sort of time zone is good for which particular part of the world. Obviously, lunchtime is really good for London, so that's very good to know. Okay, we are a couple of minutes into this now, so we've got a fair amount of the audience in. So I'm going to start going through how this session is going to work. First and foremost, my name is Kim. I'm the Outreach and Communications Coordinator here at UCL Biochemical Engineering, and it's absolutely brilliant to have you with us for the first of our virtual taster lecture series of 2025. So depending on what kind of device you're joining from, this will look different if you're on a mobile device, a tablet, or a desktop PC, desktop computer. But you have the option of changing the way it looks. So you'll notice that you have things like a chat function, reconfiguration of the speakers. You can choose to use, and there's a Q&A button as well. So if you have any kind of technical problems throughout this webinar, please let us know using the chat function. Myself and the other presenters will get to see that, and we can react. So if you can't hear us or there's any problem with the video, anything technically, please let us know. We see that straight away. We're really looking forward to your questions. You're free to post them at any point in the session, but we'll keep them until the presentations are over. But please use the Q&A function. It really helps us to package them up so we can see that, you know, if several people have asked a similar question, we can answer it more broadly. You can also ask it anonymously, and it might be that it's something easier for me to reply to you directly. So please put your questions in the Q&A box at any point in the session. And now I'm just going to go to my checklist to make sure that I cover everything. So one of the things that I just want to say is that this taster section on engineering the future of cancer therapy. If at any point the science or the engineering seems to advance, if you come and study with us, if you come and do a degree, we will take you to the level where you get this. So whether it's physics, biology, maths, part of our degree program or our postgraduate programs is that we will take you to the level where you will understand that. So we have modules particularly designed to do that. So I think that's covered everything. So what I'm going to do now is I'm going to hand over to my colleagues, Dale and Pierre, and one really important thing, nearly forgot to do this, but we're going to do a quick poll. We'd just really like to know, first of all, whether you have an offer to study with us. It just helps us know whether we have our prospective students here, or whether we've got a wider audience. It's very informative for us. So at the moment, it looks like the majority of people don't have an offer to study, which is interesting because one of the things we did was we reached out to people who have an offer and invited them along. So I can see we've got a couple of offer holders here. Great. But the vast majority of people don't have an offer to study. Really helpful. Thank you. And the next one, we just want to know if you were coming to study with us, whether it be for a degree or a postgraduate program. So this just really helps us make sure that we're reaching out to that we've reached out to an audience, what kind of audience, and to make sure that we're pitching it right, but also just out of interest. So at the moment, it looks like the vast majority of people would be more interested in studying degree, but there are a few prospective postgraduates. Thanks, that's really helpful. Appreciate you taking the time to fill that in. And now that we've done that, I'm going to hand over to Pierre and Dale. I'll turn my camera off, but I will be here in the background. Okay, so over to you, Pierre, first of all.
Yeah, hi everyone. Thanks very much for joining today. My name's Pierre. I'm a PhD here in the Department of Biochemical Engineering at UCL. My research has focused on CAR T therapies, which we'll go through in some more detail together today. But these are cellular therapies that have been modified to recognize and eliminate cancer. And my research has focused on making these drugs faster, better, and cheaper. So really focusing on the manufacturing of these drugs. And I work in close collaboration with Dale here, who focuses on something slightly different. So I'll let Dale present himself and his work.
Sure, thank you. Hi, good afternoon. Yeah, so I'm Dale Stibbs. I'm currently a postdoc working at Mervi's cell and gene therapy bioprocessing group. So just touch on my
Sorry to
background, I
interrupt you
was
there.
an
Sorry,
undergraduate at UCL.
sorry, Dale. I just want to interrupt you. You just dropped out for a moment there. If you could just repeat that from the start, it was just a little bit glitchy. Thanks.
Okay, sure. Yeah, sorry. So I'm Dale Stibbs. I'm a postdoc working with Kasim Rafiq here at UCL. So I was originally an undergraduate at UCL from 2014 to 2018 doing the MEng program. And then I moved on to do the EngD doctorate program from 2018 to 2022. And now I'm currently a postdoc. So my research mostly focuses on the scalable and continuous manufacture of viral vectors, which we'll touch on today. So I've worked on a variety of different vectors from lentivirus, which I'll discuss in the presentation, to adeno-associated viral vectors as part of my postdoc. And I've also done some work on adenoviral vaccines as well.
Okay, so with that done, I'll just share my screen and we'll jump right into today's presentation. There we go. I'll put that in full screen. Kim, can you give me a thumbs up if you can see that? All good?
Fine.
Okay, great.
It's not in presentation. That's it. Fantastic. Thank
All
you.
good? Awesome. Thank you. So again, really happy to have you guys with us here today. Really looking forward to the talk. So me and Dale will both split the presentation in two. I'll get us started and then hand over to Dale. But in today's talk, we're going to be talking about how we're trying at UCL to engineer the future of cancer therapy, specifically looking at innovations into lentiviral vector and CAR T cell manufacturing, which we'll jump into in some more detail now. So first we'll go over what are CAR T cells, what are these therapies, what do they do, what are lentiviral vectors, why do we need to focus on these things as well, and then we'll go into a little bit more into scientific detail for each of those two sections, and then we'll just conclude with a summary and conclusions, and then we'll open up the floor to any of your questions. So firstly, just discussing what are these CAR T cell therapies. So CAR stands for chimeric antigen receptor, and that is a receptor that we introduce into T cells. T cells are one of your white blood cells that are part of your immune systems, and we can genetically modify them. We introduce this CAR gene into your T cells, which then allows your T cells to recognize and eliminate cancer cells. So here in the figure, you can see you'll have a patient that's suffering from typically leukemia cancer. We can harvest some of their blood, isolate just the T cells from that blood sample, and then we genetically modify the T cells. So we put this CAR gene inside of them. We then grow the CAR T cells to numbers that are required for a dose, and then we can reinfuse those modified cells back to the same patient, where the cells can now recognize, go around the body, and eliminate the cancer inside of the patient. So just to give you a bit more information on the mechanism of action, here we have a cancer cell, let's say a leukemia cell. Now, typically, cancer is very smart. There's a number of ways that cancer can hide itself from our immune system, which is one of the reasons why cancer is so difficult to treat. But cancer cells do have receptors on their cell surface, and in this case, one of them is called CD19. And typically, your normal T cell, your normal immune system will not be able to recognize this receptor or the cancer cell itself. And therefore, your immune system fails to eliminate the cancer, causing the cancer to grow, causing the complications associated with leukemia. But here we are modifying the T cells. We're introducing this CAR gene, which is a receptor on the cell surface. And that CAR receptor is able to recognize, in this case, the CD19 receptor. And so when we give the CAR T cells back to the patient, this receptor recognizes the cancer cell, and these receptors and antigen can bind together. And when they bind together, this triggers a number of cascades, signaling cascades inside of the T cells. The DNA gets remodeled in different various ways. And the impact is the CAR T cell gets activated and releases a number of substances, such as granzymes and toxic cytokines like TNF and interferon. And these, in turn, cause the elimination of the cancer cell. So that's how CAR T cells are able to recognize, target, and eventually eliminate cancer cells in the body. So that's a little bit about how they work. And I'd like to give you a bit more of an idea of how revolutionary CAR T cells and CAR T therapies have been in the treatment landscape for cancers, particularly leukemias. So these have been around for just about over a decade. And one story we really like to share is the story of Emily Whitehead, who was just a few years of age and had terminal leukemia as a child. She tried every possible treatment option, so chemotherapy, radiotherapy, and unfortunately, everything was failing. And the doctors had told the parents, unfortunately, your daughter only has a few days to live. And it was at that moment she was the first child to ever be enrolled into a CAR T therapy clinical trial. This was back in 2012. And almost within a matter of weeks, the leukemia disappeared. The CAR T therapy, which was experimental at the time, worked, led to the elimination of her leukemia. And you can see there on the left pictures of Emily growing as a normal, healthy adult, even though she had terminal stage leukemia. So she's been 1 year cancer-free, 5 years cancer-free, 10 years cancer-free. I think she's now approaching 15 years cancer-free. So you can see that we're starting. We always have to be careful using the word cure because you never know if the disease might come back. But it certainly seems like we can start to cure some of these very severe blood cancers using therapies like CAR T cells. And on the right there, you just have a little anecdote. So once the treatment actually worked, former President Barack Obama invited Emily to the White House just to congratulate her and meet her. And Emily told the president at the time, look, I've missed a day of school for this. I wasn't allowed to tell anyone at school that I'm meeting you. Could you please write me a note? And so on the right there, you can see Barack Obama saying, please excuse Emily from school. She was with me, signed Barack Obama. And Emily Whitehead really is one of the faces of the CAR T therapy, patient advocacy at the moment, and really is a symbol of hope for all of us working in this research. So we've established what CAR T cell therapies are, how they work really well. So let's focus on why there's a need to actually improve the manufacturing of these therapies. So on the left there, you have autologous CAR T. So autologous means you have one patient, you take that one patient's cells, you manufacture them, and you have to give them back to that same patient. That's what autologous means. So these are very custom, tailor-made therapies for each patient. Each drug is made individually for each different patient. That makes manufacturing very complicated. It's like a tailor-made suit. Everyone has different cells, different blood types, and so you have to change the manufacturing process every time for every patient. And that means it's very complex to schedule the manufacturing of these. These are very ill patients. There's a number of different challenges, and when you combine all those challenges, it means that the manufacturing time takes a really long time. It takes anywhere from half a month to a full month to actually make these therapies. And because the patients are so severely ill, unfortunately, it's not uncommon to hear that patients unfortunately pass away in the waiting line. And so we can all agree that that's not good enough. And so you can see that there's clearly a need to make these therapies much faster so that we can increase the accessibility of these therapies to patients that are in need. Again, when you put all these challenges together, it means that the currently approved CAR T therapies on the market are very expensive, typically $500,000 per dose. And again, that's way too expensive. If we want to be able to treat a huge number of different patients all around the world, we have to bring that cost down. And one of the main reasons the cost is so high is because the manufacturing takes so long and it's so complicated to do. So that's why we need to focus on the manufacturing. And the first ever CAR T therapy was approved by the FDA, which means this is something that's now available on the market for people to purchase. Since 2017, only 40,000 patients have been treated. Now, that's a very small number when you consider the number of cancer patients in the world are in the hundreds of thousands, and we've only treated 40,000 in the past 8 years. So for all those reasons, we need to improve the manufacturing to make it faster, better, and cheaper so that we can get these life-saving therapies into the hands of many, many more patients for a much, much cheaper price all around the world. Now, just to give you a little bit of an idea of the actual manufacturing process, which will help us understand why this process is complicated. So going from left to right, we start with the patient. We harvest the blood and the white blood cells from the patient. We select just the T cells that we are most interested in, and then we will activate the T cells. This gives them the signal to start growing. Once they're activated, we will perform the genetic modification to insert the CAR gene. And we can do this with a number of different tools, such as lentivirus, which is what Dale's going to be focusing on. But I'm sure you're familiar with tools such as CRISPR, lipid nanoparticles. These were used in, for example, the COVID vaccine. So there's a number of different tools that we can use to modify the cells. But typically, these processes are relatively inefficient and they're very expensive. And so if just that one ingredient of the whole process is very expensive, it just drives the cost of the overall process as well. So there's a lot of room for improvement in the gene modification step, which is what Dale was going to be focusing on. And then we need to grow or expand these modified cells. So generate enough numbers to actually get to a therapeutic dose. And now the cells we get from patients are of poor quality. These are very ill patients, and growing them can take a long time. And it's usually the longest phase of manufacturing, which again, takes a lot of time. It draws out the process. It means the patients have to wait longer, and the longer the manufacturing, the more expensive it is. So we need to grow these cells faster. That's what I'll be focusing on. So just to summarize, to make CAR T therapies more accessible, we need to make lentivirus cheaper and more efficient to modify the cells. And then once we've modified them, we need to find ways to grow them faster and cheaper, which is something I'll focus on in the next part of the presentation. So I'll hand it over now to Dale to talk about lentiviral vectors.
Thanks, Pierre. So as Pierre's shown on that slide with the manufacturing process, genetically modifying the cells is a really critical step in the CAR T manufacturing process. There's quite a few different approaches we can take to this, but the dominant one at the moment is using a viral vector, and specifically in our manufacturing process and in a lot of the commercial FDA-approved manufacturing processes, they're using a lentiviral vector. So I think it's worth just discussing how these lentiviral vectors actually work. So the process whereby you use a viral vector to genetically modify the cells so they can express that CAR on the surface is known as transduction. So if we look at the schematic on the screen, the first stage is really that the vector binds to the cell surface receptor on the isolated T cells. So at this point, you've isolated your T cells, and then you put your vector and T cells in contact with each other. The lentiviral vector fuses its envelope with the target cell membrane, and then the viral components are then able to enter the cell. In stage 3 of the process, the viral RNA is then reverse transcribed into DNA. And then that DNA is actively transported across the nuclear membrane, and then enzymes integrate it into the chromosomes to achieve stable gene expression. So the biggest advantage of lentiviral vectors is that the CAR you're expressing on the surface is stably expressed for a long period of time. It's not transient, it's not there for a few hours or a few days, it's stably expressed and it's continuously there on the surface of your cell. So at the end of this transduction process, you modify cells and now the CAR receptor on the surface, and you're ready to carry on with the process that Pierre described previously. If we can go to the next slide, please. So the particular type of lentiviral vector that we use is a modified version of the HIV-1 virus. So what we found is that we're able to modify this virus to function as a tool that can deliver our gene, our CAR gene, to the cells so that they're expressed on the surface. So lentiviral vectors have been developed through an iterative process that has mostly focused on making them safe to use. So we're at the point where we have 3 generations of lentiviral vectors. The first one was essentially a full HIV-1 genome with very minimal modifications, and this presented quite a high risk of replication, and it was essentially quite dangerous. So we then found that you can remove some of the genes and it can still function as a vector. And by removing some of these genes, you're able to increase the safety profile of the lentiviral vector. So in the second generation, they removed 4 genes, which then increased the safety of the vector. And then finally, in the third generation, we were able to remove another 2 genes and find that it still perfectly functions as a vector, and it's able to do that gene delivery, but it's much safer for both the patients and the people manufacturing the therapies. So taking modification that's really the function of lentiviral vectors is that we've modified that so that when on the previous slide, when I the cell and the vector, there's a binding to the cell surface receptors. We've modified the lentiviral vector so that it's able to bind with a very broad spectrum of cells, which is really important for manufacturing cell therapies. So that process is known as pseudotyping, and we found that a particular type of protein that we put on the surface of the cell allows us to transduce a very wide range of cells, and it also appears to increase the stability of the vector, which is really important from a manufacturing point of view. So vectors are often quite unstable, and that's very challenging to use and to manufacture. So if we can move to the next slide, please.
Yeah, Dale, just there's a little bit of misconnection. So maybe just if you try turning your camera off, it will help with the audio.
Yep, sure. Do we have the next slide? I can't quite see it yet. Oh, yeah, got it. So these lentiviral vectors, they're roughly spherical molecules. So they have a diameter between about 80 and 120 nanometers. They're also negatively charged. So this is really useful for purifying them. So when we come to purify viral vectors, we typically use anion exchange chromatography, whereby we can separate the negative viral vectors from positively charged impurities. So as we can see in the red box that I've highlighted on the figure, this is the envelope protein on the surface. So it's this that binds with the cell. So we found that a particular type of envelope protein known as VSVG, this expands the tropism of the LVs. So this allows them to transduce a very wide range of cells, from those T cells that Pierre described to neurons and stem cells. And as I said, it also increases the stability of the vector, allowing us to manufacture them at very large scale. However, there are some drawbacks to this particular type of envelope protein. It's cytotoxic, which does present challenges for manufacturing them, and it's also it's quite difficult to manufacture them because essentially it's cytotoxic to the producer cell. So if we can go to the next slide, please. So as Pierre mentioned, one of the real challenges in CAR-T manufacturing is the cost of the vector. So there's a few different approaches that you can take to manufacture lentiviral vectors. One is known as transient transfection. So this is by far the most widely used approach. Here, we use a chemical reagent to introduce the plasmids encoding for the lentiviral vector genes into cells, producer cells that manufacture the vector. So typically we use a cell known as HEC293 T cells. These are often adherent, so they need a surface to attach to be able to grow. And the real drawbacks to this process are the costs of the plasmid DNA themselves and the transfection reagent. So this represents a very large proportion of the overall CAR-T manufacturing cost. Transgene transfection also has some drawbacks in that it can result in quite high levels of batch-to-batch variability. However, this is a very well-established approach. You can produce lentiviral vectors at extremely high titers, and it's also a very flexible approach. So if you see these three plasmids on the screen, if you wanted to switch to using a different envelope protein, so the one in the middle, or you wanted to use a different transgene, so instead of expressing a CAR, you wanted to express GFP, for example, that's something that you're able to do because it's a very flexible process. If we can go to the next slide, please. So as I mentioned previously, the cells that we often use to make lentiviral vectors are adherent. So there are really two different ways you can grow these cells and produce the vector. So one is using flasks. So this is on the left-hand image. So a flask is a really cheap and easy to use way of producing viral vectors. However, it does have some drawbacks in that you can't control any critical process parameters. So for example, the pH in this flask cannot be controlled. And also scaling becomes quite difficult. So if you wanted to take your process and scale it up to make a huge amount of lentiviral vectors for your CAR-T manufacturing process, that becomes really challenging. And what you often have to do is use these stacks, which are in the middle. So while these may be able to produce sufficient amounts of vector for kind of lab scale manufacturing processes, for these clinical manufacturing processes, you often need to use something that's much more scalable. So we often use a bioreactor. So a bioreactor allows for and provides much more precise control of process parameters, particularly the culture pH and bulk oxygen. It's also much more scalable. So the bioreactor can often produce vector at scales of 10 or 100 times larger than what a flask is capable of. The bioreactor, of course, has some drawbacks in that it has a relatively high capital expenditure, and it also requires trained users to be able to operate it. So yeah, if we can move to the next slide, please. So I'm just going to touch on some of the work we've done here at UCL on lentiviral vector manufacturing. So we've used an alternative approach for transient transfection. So we've used a cell that's been modified to continuously produce the lentiviral vector. So that transfection process that I touched on previously, we don't use that. We don't need the transfection reagent, and we also don't need the plasmid DNA. So this reduces a huge amount of the cost of manufacturing those lentiviral vectors. So these slides do have a few drawbacks in that they're very challenging to manufacture. It's a very time-consuming process, can often take several months. And they're also, compared to transfection, the titers are often lower. Sometimes they can be 10 times lower than what you would get from a transfection-based process. But this has the potential to really revolutionize lentiviral vector manufacturing and significantly reduce the costs. So I'll briefly touch on some work we've done here at UCL. So we've used one of these cell lines and established a continuous manufacturing process for producing these LVs. So we first started off by establishing a process in flasks, just like the ones we saw on an earlier slide. And this process, we first looked at the impact of seeding density. So as you scale your process up to very large scales, having just enough cells to start your process to seed the vessels becomes very problematic. So we ran a study to try and minimize seeding density. So if we move to the next slide. So what we found, what the data showed is that if we seed at 30, 40, or 50,000 cells per centimeter squared, we're seeing similar levels of LV. So what we decided to do was use the minimum seeding density. So in this case, it was 30,000 cells per centimeter squared to try and make the process as easy to scale as possible. So once we had this process in the flask, we then moved on to our bioreactor. So we used the same one that was shown on the previous slide. So this is known as a fixed bed bioreactor, and it's specifically designed to expand adherent cells. If we can go to the next slide, please. So we ran a number of different processes in this bioreactor. First, we started off by transferring the process from the flask to the bioreactor, and we did that by keeping the seeding density the same and also keeping the medium exchange rate the same. So this is how frequently are we changing the media that provides nutrients to the cells. So in the first process, we were changing it once per day. And then if we can go to the next slide, please. So then we carried out a number of studies to look at critical parameters on lentiviral vector manufacturing. So we first looked at the impact of changing the culture pH. So we know that pH is important for lentiviral vectors, and a lower pH often results in higher titers. So we found that lowering the pH slightly did increase our yields. And then the second thing we found was that by increasing the medium exchange rate, so how often are we changing the media, replenishing the media, we can also increase our titers that way. And what we think is happening is that because the vector is very unstable at 37 degrees, which is the temperature we need to grow the cells at, by reducing the amount of time the vector is spending in the bioreactor, we can increase the amount we produce overall by removing it from that 37-degree environment and putting it in a cooler environment, either at room temperature or in a fridge. So this slide that we're seeing on screen here just shows the variability of our process. So overall, through all this process development, we were able to increase our yields about threefold. And one thing that we saw, which is a really valuable the value of this approach, is that the variability we see across the processes is very low. So it's a very consistent approach of manufacturing lentiviral vector at a very large scale. Yep, so I think that's it for the lenti work. I can hand back to Pierre now.
Dale just covered, how do we improve lentiviral vectors, which is critical to actually modify the T cells into CAR T cells. So now we actually have CAR T cells, we need to grow them. So this is the same slide I showed you at the beginning, but we're just going to focus on the challenge, which is expanding them or growing them, which currently takes too long. So we need to accelerate the growth of these modified CAR T cells. So the approach of my research has been, like Dale, to try and use bioreactors. So you have a bioreactor here on the left, where it's a container which you can control the mixing, the pH, the oxygenation, the temperature. You can more or less control everything you like. And so the yellow liquid there represents the CAR T cells that are growing in a liquid. And we've tried to use perfusion to improve the growth of these cells. So what is perfusion? Perfusion is the continuous exchange of the culture medium. Culture medium is the liquid in which cells grow in. And by continuously exchanging this liquid, we're removing toxic waste products like lactate and ammonia. And at the same time, we're replenishing nutrients that the cells need to grow. So we have this bioreactor here, and on the right, you have a perfusion filter device, which is this fancy tower on the side of the bioreactor. With pumps, we can pump the cell in and out of the reactor across this filter. And what this filter is used for is to separate the liquid from the cells. The liquid will go to the waste. We call this spent medium. That's got all the nasty stuff you don't like, the toxic lactate, ammonia, that goes to waste. And at the same time, we're replenishing the bioreactor with fresh medium, which has your glucose, your glutamine, all the nutrients the cells need to grow. And so we know perfusion is a process that can improve cellular growth. It's been used in the production of different biopharmaceutical products for many decades. But for CAR T therapy manufacturing, it's still too early and it's still an unoptimized process. So I was looking at, can we improve this perfusion process to maximize growth for CAR T cells specifically? So can we optimize or improve this perfusion to maximize the CAR T cell growth? So here's an overview of the entire process. So on the left there, you have some of those steps we'd covered previously. So first, the cells that I start with are frozen. So first you thaw them into liquid, you then activate them, which gives them that signal to grow. We then modify them with some of the lentivirus that Dale produces. He then gives it to me, I use it. We modify the cells, we grow them for a couple of days, and then on day 5, D5, we can put the cells into the perfusion bioreactor. And we grow them for 7 days, and we tested 17 combinations, changing and tweaking some of those perfusion-related parameters to try and see if there's any impact and if we can improve the growth even better. So here you have another representation of the bioreactor. This is a real life photo. So you can see on the left, the CAR T cells are in the liquid, and on the right, you have the perfusion device that allows you to do this exchange of liquids. And this is the actual machine. So you can put 24 of those little bioreactors into this bigger machine, which allows you to do 24 different experiments all at the same time. So I can test loads of different parameters, loads of different experiments all at the same time with this big platform. So let's look at the data. So on the left here, you've got 17 different growth curves, 17 lines that represent how the cells grew in 17 different experiments. And in each experiment, we changed some of those parameters. Sometimes we made it lower, higher, we mixed the two to try and see if there was an impact on the cell growth. So as you can see on the left, there was definitely a big impact on cell growth. Our historical process, our old process, had no perfusion, and that's represented by the black arrow at the bottom there. All the other colored growth curves are perfusion growth curves. So you can see that just by doing these different perfusion experiments, we've drastically increased the number of cells and their growth in the bioreactor. In the best condition, we almost multiplied by 5 the total number of CAR T cells in the bioreactor. And we look at different things. So on the right, you have the CAR T cell viability. So out of 100 cells, how many of them are actually healthy? How many of them are actually alive? So we track that over the 7 days in the bioreactor, and you can see that generally we're above 90%, which is a really good number we're really happy with. So over the week, even though we're changing different parameters in perfusion, generally the viability stays really high. So the vast majority of the cells in the experiments are still alive, which is something you definitely want to see happening. And then we can use, which is something we teach at UCL in the master's and at the PhD level as well. We use these different statistical approaches to try and identify the sweet spot. So I told you we were changing these different parameters at the same time. So we need to find the sweet spots that maximizes the growth at the best. So you can see these 3 plots. Each of them come from 3 different donors. So I used cells from 3 different healthy people, and I put them in my bioreactor, and then we tracked the growth. The redder the graph, the more growth there was. So that's where we want to be. We want these red hot regions, which represent the fastest, best growth. And with these statistical techniques and analyses, we can identify the best region and the best combination of parameters in our experiments to maximize the growth, even for different donors. So you can see there's some slight differences for each of the 3 donors, but generally to maximize growth, you need it to be in that top left. So that way we found the optimal recipe, if you like, to maximize CAR T cell growth, even for different donors. And just to translate this into why this is important clinically and for patients. So on the left, you have our historical process, the dotted line with no perfusion. You can see the cells didn't grow particularly well. And then in blue, you have the best perfusion condition, where we multiplied almost by 5 the cell growth. So if we look at the table on the right, the total doses produced in 7 days with no perfusion, we only made 1 dose in 1 week. But with optimized perfusion, we made 4.5 doses in the same amount of time. So we made many more doses in the same amount of time. Importantly, the most important is how long does it take us to get to that first dose? So in the far right column, the time to reach that first dose with no perfusion, it takes a whole 7 days to get to the first dose. But with optimizing perfusion, we can drop that down to 3 days. So we're reducing by over 50% the time required to reach that first dose. And that's so, so important. If you remember, I told you, unfortunately, sometimes patients pass away waiting for these therapies to be manufactured because it just takes too long. So by optimizing perfusion, if we can reduce the time to grow the cells by over 50%, we can make them faster, which will make them cheaper, and we can get them to patients in need much, much quicker. And then, I grow these cells in these bioreactors. We need to make sure, do they actually do what they're supposed to do, which is eliminate leukemia cancer cells. So at the end of the experiments, we take the CAR T cells from those bioreactors and we mix them with cancer cells to assess whether they can effectively kill them. So in that middle graph there, we're looking at cytotoxicity, and the purple triangle line that goes upwards, these are non-modified T cells. So they're not able to recognize the cancer. So the number of cancer cells just keeps growing, keeps growing, keeps growing. The black line, which drops down, these are the CAR T cells that can recognize the cancer and eventually eliminate them. So that's why we see the number of cancer cells dropping down. So here we're showing the CAR T cells are effectively recognizing and eliminating the cancer cells. And then on the far right, we investigate, are the CAR T cells actually releasing these toxic substances? So on the far left there, you have that mechanism of action I showed you. So one of the substances is tumor necrosis factor, TNF. So on the far right, we just measure, do they actually release this? And the CAR T cells from perfusion definitely do release this cytotoxic cytokine. Whereas the non-modified, the normal T cells, they don't do that because they can't recognize the cancer cells. And then lastly, just to show you what this actually looks like under the microscope. So we harvest the CAR T cells and we put them together with fluorescent green cancer leukemia cells. On the left side, we're looking at non-modified T cells. These cells are in gray. They cannot recognize the green cancer. This is what happens in a normal patient's body. So the cancer that's green just keeps growing, keeps growing, keeps growing. So that's what's happening in an untreated patient. On the right here, we're looking at the perfusion CAR T cells. We've modified these cells to recognize the green cancer. And so over the space of 2 days, you can see the vast difference. The cells are recognizing and then attacking the green cells. And then within the same space of time, you've effectively got no more green cancer cells left. So just giving you a visual representation that our CAR T cells are indeed doing their job. They are functional and they are able to eliminate the cancer cells. So that brings us to the final slide where we'll just try and summarize and conclude some of the things we've gone over. So I'll just summarize for Dale as well here. Lentivirus, firstly, is a critical tool for genetically modifying the T cells into CAR T cells. But this can be, for a number of reasons, a very expensive process, and the amount of virus that we can generate is typically relatively low. And so using some of the methods and approaches that Dale's explored in his research, such as continuous lentivirus manufacturing, we can really reduce the costs to make this lentivirus and also lower the batch-to-batch variability. So each time you do this, it's going to be quite consistent. Once you've modified the CAR T cells, we need to grow them. And so we've shown CAR T cells can save patients, but the amount of time it takes to grow the CAR T cells is the longest phase of manufacturing. And the longer something takes, the more expensive it's going to be to make. And so we've shown that by using perfusion and optimizing perfusion, we can accelerate the growth of these CAR T cells. We can generate a clinical dose in more than half the time. And so by reducing the overall time, we're reducing the overall costs, and we can get these therapies cheaper to patients faster. And so hopefully you can see both from my research and Dale's that designing more efficient manufacturing methods, which is what our whole department in UCL is all about at Biochemical Engineering, it's crucial to improve these processes to increase the accessibility of these life-saving cell and gene therapies to make them better, faster, and cheaper, and get them to many more patients that are in need. So we hope you've enjoyed that presentation, and I think we'll open the floor up to any questions.
That's fantastic. Thank you very much. I can see we've had a bevy of questions come through. So what I'm going to do is I'm going to give Pierre and Dale a chance to look through them. I think there are a couple which touch on the same area, and possibly some, because they were submitted early in the presentation, they might have even been covered. So it's worth giving Pierre and Dale a chance to read through them to see how they're going to approach those. One thing I just want to say is that we've had a couple of questions about degrees at biochemical engineering, the difference between biochemical engineering and other subjects. Myself and Dr. Jack Jeffries will be online. We've got 2 sessions coming up on the 5th and 6th of May. We're doing one in the afternoon, one in the morning, so hopefully there'll be a time zone that works for you. If you want to find out about our degrees, if you sign up, and I'll put a link in the chat in a moment for that, or you can go to the front page of our website. We can talk to you about our degrees, the content of them, entry requirements, what modules go into them, what kind of composition. There's even a pre-recorded video which Jack did talking about that. So watch the video that he did. He goes through our programs in details, explains the difference between chemical and biochemical engineering, which comes up quite a lot. But do come along, and we'll talk to you about careers in biochemical engineering, what our graduates go on to do, and a whole other range of things, including what kind of labs we have and all that, and anything which you want to ask about. But come along on the 5th and 6th of May, because that's when we can go into detail on that. Unfortunately, we don't have time to do that now. Pierre, Dale, if you want to kick off, I'll let you go. We've got probably a good 10 minutes to go through questions, so we'll get through as many as we can. I can see there are quite a few.
Yeah, some of these are really good actually, so that's good. Dale, I'm going to start off by throwing one at you. So we have one question here about lentivirus, which is, what are the benefits of lentiviral gene modification compared to other forms of gene modifications?
Yeah, so first, to compare lentiviruses to other types of viral vectors. So one of the main benefits of lentiviral vectors is that you're able to transduce both dividing and non-dividing cells. So if you go back to that diagram that I showed earlier, when the DNA is actively transported across the nuclear membrane and then integrated into the cells. So other types of vectors are not able to do that process. So that's why lentiviral vectors are able to do that for dividing and non-dividing cells. Lentiviral vectors are also, as I mentioned in the presentation, able to stably integrate the gene into the chromosomes, and it's expressed long term, which other types of viral vectors don't necessarily provide. Then to compare them to non-viral approaches, we've done quite a bit of work here at UCL on non-viral approaches, and it's a very promising way of gene modifying the cells. However, at the moment, the efficiencies, so the number of cells or the percentage of cells at the end that are expressing your CAR is typically a bit lower than what we're seeing with vector. So vector does have plenty of drawbacks, but at the moment it's probably the best way that we're able to modify these cells in CAR-T manufacturing.
There's another question here, Dale, which you can probably answer. So some cells are adherent, which means they need to grow on the surface of something like plastic. Other cells are suspension cells. They can just float in liquid. So the question here is, in what situation would you move over to suspension cells rather than using adherent cells? Why would you want to do that?
So you would always, for viral manufacturing, we would probably always want to try and use suspension cells, because we're able to produce the vector, whether it's lentivirus or another type of vector, at the scales that we need to either manufacture these therapies in, such as CAR-T, or to use the vector as a therapy itself. There was one reason why we didn't do that in this case, and that was, if you remember, I said that typically stable cell lines generate less lentivirus on a per cell basis than transfection. So typically this can be about 10 times lower. So if we were to try and adapt these cells to suspension, the literature shows that that is associated with a further reduction in the titers. So at that point, our process on a per cell basis would be producing potentially 100 times less vector than a transfection-based process, which probably wouldn't make it competitive, and the titers would just be too low. So in this specific case where we already had a cell line that was adherent, if we were to start again, perhaps we would make a suspension cell line, but it was already adherent. We didn't want to reduce the titers any further. So what we tried to find was an approach of producing vector at a much larger scale using that adherent cell line, and that was the reason we went for the fixed bed bioreactor, because it does allow you to expand adherent cells. But adherent cell manufacturing always poses problems with regards to scalability. And these types of bioreactors Pierre discussed in this work, it would be our preference to always use these types of stirred tank bioreactors.
So we also have quite a few questions about CAR T therapies themselves. So I think one which would be good to start off with is, can you explain why CAR T cell therapy manufacturing is so difficult? Why is it so difficult to create these therapies? So one of the key reasons is it's just so new. These have been around maybe 10, 15 years. And to do this at scale, to treat hundreds of thousands of people, is very challenging. So if you look on Google, if you look at the photo of the first ever computer was the size of a room, and it's like 10 scientists using it. And now we each have a computer in our back pockets. So that's what we're trying to do with CAR T therapy manufacturing. It's just very new, it's very manual, everything's very expensive, everything's very new. So by improving the manufacturing, just like the computers have managed to get into our back pockets, we'll be able to improve these processes to make sure we can make CAR T therapies cheaper, faster, and better. Then I also have a couple of different questions that are hinting at the same theme, which is, can we use CAR T therapies for different diseases, different cancers? Why are they targeting leukemias at the moment? So right now, leukemia is blood cancer. And one of the reasons why CAR T therapies currently target leukemia is it's relatively easy to get those CAR T therapies everywhere in the blood. You just inject the therapy in the vein, it goes all around your blood circulation, and it can target the cancer everywhere. So that's why right now it's targeting these blood cancers. Other cancers, like solid tumors, brain tumor, liver tumors, those are much more challenging. Firstly, to get to, how do we get the cells there? You probably need to have surgery to do that. And these tumors are very hostile environments. Once you put the cells in the tumor is very intelligent. It will actually attack the CAR T therapies. But there's a lot of work going on now. Now we've shown CAR T therapies work for liquid blood cancers. There's a huge amount of research, there's a lot ongoing at UCL, to target the solid tumors, because there's many, many more types of solid tumors out in the world than there are of blood cancers. And just on the subject of diseases, we need to think bigger. It's not just cancer. So a lot of really promising research is coming out at the moment for autoimmune diseases, such as lupus, for example. And there we're seeing really, really encouraging results to treat these patients that have these autoimmune diseases, but also other diseases like, for example, graft versus host rejection. If you get implanted a new organ, your body will attack it. Well, it seems that CAR T cells could actually help protect your new organ from your body, for example. So there's a huge amount of research, and hopefully we'll be able to apply these to many different types of diseases. Let's see what else we have.
So just saying that we're coming to the last 5 minutes, so we'll keep going until the very end. But I just wanted to also come in, give Pierre and Dale just a chance to go through the questions, have a quick scan in case there's anything they really want to cover again, because we've had loads more come in. But also, I was going to say we've got 4 more sessions coming up, and we're going to be covering vaccine manufacture. We're also doing one tomorrow, which is going to be on lab-grown meats. We're going to be focusing, and that's when we were talking about, can you actually make dodo nuggets? So you're taking an animal that's actually extinct. Can we get the DNA from that? Can we grow it? What kind of structure do we need? Could we make a nugget versus like an actual leg of a dinosaur or something? It should be quite grotesque and really good fun tomorrow. But we've got another series, 4 of these coming up. So do sign up for any others that are coming along. But right, over to you, Pierre. Dale, maybe 2 more questions.
Yeah, I'll just take one and I'll ask Dale one. So In mediating cytotoxicity of CAR T cells and reducing off-target effects, are these part of your concerns in biochemical engineering sectors? So once you administer these CAR T cells, how do you control off-target undesirable effects? So that's a really good question. And that is definitely something that can happen. Sometimes patients do get side effects like fevers and things like that, which we can control and treat. Because we're really focused we're in an engineering department, we're really focused on the manufacturing. So in our day-to-day research, we're less focused on what actually happens when these therapies go into the patients. That's what the clinicians and clinical researchers focus on. But because there's a close link between what we make and what the doctors use, a lot of our research is done in collaboration with clinicians. And so we work with them to try and make CAR T therapies that will result in less off-target effects. So we try and work with clinicians wherever possible. And maybe last one for Dale. How do you make lentivirus for CAR T manufacturing and keep it or store it in different weathers and temperatures? What can you say about storing and transporting these viral vectors, Dale?
Yeah, so we've done quite a lot of research on the stability of the vector. So we know that typically it's stored at very cool temperatures. So essentially it's frozen. That helps preserve its functionality. So typically the vector is susceptible to high temperatures. So as I mentioned in the work, typically when you set 37 degrees, that's when it's it's very unstable at those temperatures. So we want to try and put it in cooler temperatures, and this is typically done by freezing it. And when you're freezing vector, typically you're worried about ice formation, which can damage the vector. So we often see losses of vector as a result of freezing and thawing them. So there's plenty of work that's been done on trying to minimize the effects of freezing and the losses associated with freezing and thawing. I can maybe take one more if we have time. It says, could it be possible to one day use lentiviruses to create CAR T cells within the body? So yeah, there's quite a large area of research focused on using not just lentivirus, but also other gene delivery approaches such as mRNA to try and eliminate essentially everything we spoke about today pretty much and just deliver gene modified cells within the patient. So this is typically referred to as in vivo CAR T. I don't know if Pierre can comment anymore, but this is at quite an early stage. But yeah, this is an active area at the moment. And then, yeah, I can maybe just take one more in the last few minutes. Why is plasmid DNA so expensive? So yeah, the cost of lentiviral vector manufacturing are high because of the high cost of the plasmid DNA and also the transfection reagent. So typically we use a transfection reagent known as PEI. It's a cationic polymer, and this is extremely expensive to buy at particularly when you're producing virus at a very large scale. That's when it becomes very expensive. And there's kind of an issue that you end up with more and more manufacturing processes. So you need a process to produce your CAR T. You also need a process to produce your lentivirus. You also then need a process to produce your plasmid DNA. So you're sort of stacking manufacturing on top of manufacturing, and that's where the costs are sort of incurred.
Fantastic. Thank you very much. I just want to say a huge thank you to everyone who joined us. Those were really, really good questions. I did say to Pierre and Dale beforehand, before they came along to this, you'll get some really, really good questions in this that will make you think quite hard. And I can see that they did get some really good questions. Thank you very much. That was brilliant. Listen, it's come up a couple of times. People have asked about the difference between biochemical engineering and other subjects. Please join us online on the 5th and 6th. I've put links to sign up to that, but I'll be with Jack Jeffries, and we can talk in loads of detail about the composition of our programs, what the difference between one degree and another is, what kind of careers people go on to do. And I just also want to mention that Dale did our degree program as well, so Dale's come through doing a degree with us. Pierre did the MSc Manufacturing Commercialization of Stem Cell and Gene Therapies, which is the longest subject title of any program at UCL officially. It's an incredible degree. So if you want to find out more about studying with us, join us for the degree talk on 5th and 6th. And if you want to find out about any more areas of research, please join us for one of our other sessions. Also, I'll send a link to all the recordings anyway, so if you can't join us for the rest of the week, I will send links to it. But I just want to finish off by thanking Pierre and Dale. That was fantastic, really, really great, and a huge thank you to everyone who took the time to join us today. And anyone who's watching the recording, thank you for watching this, watching back. It's been great to have you with us. Hope you have a fantastic rest of the week. Goodbye. You're very welcome. Some nice, kind words in the comments. Thank you very much. Goodbye, everybody.